builds forests, it never builds up big biomass. Because, let's say, bacteria take care of the phytoplankton, the zooplankton, the food chain is taking care of that. And at the end of the day, most of the CO2 ends up in the atmosphere again. Now, if you would understand heterotrophic bacteria and their key role in carbon cycling, you might interact or at least understand how much of the biomass could potentially be exported over here to the sediments because it is not immediately remineralized by bacterial plankton. So that's our justification. It's all about biogeochemistry, large element cycles. But what we are interested from now on is mostly the utilization of polysaccharides, because a major part of the biomass of phytoplankton consists of polysaccharides. You could roughly say 1/2, but that really depends. It can be up to 80%, can only be 20%. It depends on the type of phytoplankton. Now, if we go to the next slide, this is now the first microscopic image. What it shows you is a Ketoceros assemblage. So several cells of Ketoceros. You see, this is a diatom. You see the nucleus, big eukaryotic nucleus in the middle. The Ketoceros were stained with the lectin. So you also see the spines over there. And you also see that this colony of Ketoceros is not doing very well. It's in a die-off phase, certain cells already have disintegrated. And we put that Ketoceros first on a filter in a way that also the bacteria attached and close to the Ketoceros cell got on the filter. It's a 0.2 micrometer pore size filter. And on that we collected the moribund Ketoceros cells and particular bacteria. The green ones are now Bacteroidetes, or in the marine environment, flavobacteria. I'll come back to the taxonomy later on, but one particular type of heterotrophic bacteria that are closely linked both to the surface but also to the surrounding of dying diatom cells. Now, why is it now necessary that we even consider doing omics of all cells? And the reason for that needs to be summarized for a broader audience. I'm trying to address marine biologists or marine scientists as a broader audience today. Therefore, I need to explain that cultivation is first of all cumbersome. It needs special techniques like dilution culture. And this slide over here shows you how from seawater you can dilute and ultimately get maybe a single cell or a few cells into a vial in which, if you provide a suitable medium, the cells would grow. On average, cultivation efficiency is never 100%. It is variable from place to place, from time point to time point, but overall, the efficiency might also be only in the range of a few percent, maybe 10%, maybe even less. So not everything which is out there, the microbiome consists of potentially thousands of species. Not everything has ever been brought into pure culture. And that's the reason why we need to consider comparative genomics of suitable isolates that are necessary for an understanding in the laboratory, but also going to the field with metagenomic, microbiome-based tools. Now, let us shortly go to Helgoland. Most of you know that this is an island about 40 kilometers from the coastline in the southern North Sea, in the German Bight. And some 20 years ago, we got a first isolate. At that time, we called that Gramella forsythii, Kabeltonne KT 0803. So an isolate that we got in March. The plankton isolate of the North Sea close to the island Helgoland was subsequently classified as a member of the Bacteroidetes. Bacteroides is the dominant bacterium in your gut, so a typical gram-negative heterotrophic bacterium. And the marine class of Bacteroidetes is called Flavobacteria. This particular genome was then sequenced ad linear in our network of excellence. So that was the year 2004. It was subsequently annotated by Marga Bauer and others and published in Environmental Microbiology in 2006. It is by now a model for polysaccharide degrading marine heterotrophic bacteria. It really turned into a model over the last 15 years. To just reiterate, the marine flavobacteria are a sister group of what all of us have as a dominant bacterium in the gut. So heterotrophs with a particular setup, two membranes, for example. This is a particular monophyletic group. Just go down. One more. A quick look, also still in the introduction, into the genome. The genome had a size of about 3.8 megabase pairs, and it consisted of roughly 3,500, 3,800 open reading frames, of which one particular thing is noticeable. And that is there's a lot of transporters of a ton B dependent outer membrane type and a lot of glycosid hydrolases. And those things are co-locating. At that time, we did not have a name for that, but later on in this talk, I will refer to those loci in the genomes as polysaccharide utilization loci. These are co-localized transporters and glycosid hydrolases co-localized at a locus in the genomes of gram-negative bacteria, in particular in a canonical type in the Bacteroidetes. If we now look into a typical Helgoland spring situation, I would like to explain that the textbook setup that we would expect is a phytoplankton bloom, which is dependent on increasing day length, increasing temperature, mixing. And then in spring, I pointed out March, April, maybe early May, there might be one or several phytoplankton blooms, dominated in temperate to polar regions by diatoms. They release as active cells, but in particular as dying cells, a lot of organic matter in a dissolved and particulate form. And this is now food for the heterotrophic bacteria, and with a time lag of 2 to 4 weeks, according to textbook, you would expect a bloom of heterotrophic bacteria mineralizing the organic matter back to CO2 and to everything else. That you can study now with techniques like 16S tag sequencing, in situ hybridizations, but you can also use genomics, in particular metagenomics. Now, once again, a short map so that you get the situation. It is still a shelf sea where we are at. The whole, that's a North Sea is a shelf sea. It's not very deep. We are talking about a water depth of 7 to 10 meters according to tide. And the sample is exactly taken between those two islands. There's this counterclockwise currents that goes this way. And you have, I would say almost, the proxy is we have open North Sea conditions over there. The big thing about an observatory is always it has a legacy. So Helgoland was traded versus Zanzibar with the British in 1888, if I recall it. And ever since, there is a biological station on that island. And that biological station has small ships that permanently go to the Kabeltonne, which is a long time observatory with a, let's say, history of more than 100 years. There's nice recordings also on bacteria and zooplankton for more than 50 years. There's a recording that the temperature has raised for more than 1 degree over the last couple of decades. So this is experiencing global change like Earth does. And the big thing is we can relatively easily get lots of water into well-equipped laboratories and do size fractionated filtration. Now, this size fractionated filtration is then the basis for a combination where we try to get a perception of the system, of the ecosystem, by data integration. Metagenomic data, metatranscriptomic data, metaproteomic. The title of this presentation already indicates that, in particular, a long-term collaboration that also lasts now for 20 years between the MPI in Bremen and the University of Greifswald is important to do both, not only the genomes and the metagenomes, but also the proteomes and the metaproteomes. You also have a look at diversity. I'm a taxonomist by training. If we now look at the general setup, and you need to recall that because otherwise you will lose, I would already apologize right now, a lot of the complexity that I will have in my description of the heterotrophic bacteria. Now, the first and most important thing is, for certain applications like proteomics, we still need dozens of liters, maybe no longer hundreds of liters, but we need a lot of water. For other applications, we only need a liter. For microscopy, we might only need 10 milliliters or so. But the most important thing is we do fractionated filtration. Whatever comes to the laboratory, and this is the back wall of the Biologische Anstalt Helgoland, 300 liters of water, first goes through a 10 micrometer filter to screen large particles, then goes through a 3 micrometer filter, and then is collected on 0.2 micrometer. And in this size range, 3 to 0.2 micrometer, most of the results that I'm reporting today will be situated. So we disintegrate the full complexity into a window where we look primarily into the heterotrophic. That said, we now go to the Spring Bloom 2009, which was the first one that we examined with a big campaign on Helgoland. So we always go there for about 2 to 3 months. We are utilizing the guest laboratories over there, and we rely a lot on the hospitality of the people at Biological Anstalt Helgoland, led by Karen Wilcher, but in particular Antje Wichels and Gunnar Gerd. Some of you might know them, long-term members of the Biologische Anstalt Helgoland, our collaborators over there. There was a diatom bloom dominated by Rhizosolenia. You see that over there. And we have then the typical situation. This is the chlorophyll values over there, going up to 30 milligrams per cubic meter. You see that barely, but you see it. And black, this is the number of cells, mostly heterotrophic bacteria. And you see this typical offset in terms of 2 to 4 weeks. It's in this particular case, end of March for the phytoplankton bloom, and then something like end of April for the dominant bacterioplankton. That's the situation where we do the investigation. That's our window. This is the full year, and you see that there's also a summer and a fall situation. Additional blooms follow up, usually 2 to 3 well-defined blooms. But that's the 2009 situation, and the 2009 situation is perfect because it was a deep winter minimum of phytoplankton and an explosion that was very discrete. If we now look into the taxonomy of the bacteria, so we start to look into that window from 0.2 to 3 micrometer and look for heterotrophic bacteria. A winter community has lots of alpha proteobacteria, and then the moment there is a phytoplankton bloom, you wait a bit, and with the maximum of bacteria, one particular group, the Bacteroidetes here in yellow, take over, and they are followed by some gamma proteobacteria, but overall in relative abundance. So this is relative abundance percent of all cells. The alphas go down, and the Bacteroidetes take over. This is now the window in which we did the Spring 2009 examination. More interestingly, within the group of Bacteroidetes, but also within the group of gamma proteobacteria, there is a very rapid, most likely substrate-driven succession on the level of genera. So particular species take over. They are in genera referred to as, for example, Formosa or Polaribacter. Those particular genera become dominant up to 20, sometimes 30% of all cells. And these particular genera collapse again. So they grow into a certain density and then mortality kicks in. This might be phage-based or grazing-based mortality. Now, this tight succession of different genera, some of them, if something is called NS5, we don't have a single isolate at this time. If it is Formosa, it's close to a cultured Formosa. Rhyneikea is also a cultured, Polaribacter is cultured. So there is a relatively, I would say, flat situation in the winter. And with the onset of a massive input of phytoplankton biomass, there's a tight succession of different genera. Is this pattern recurrent? That was a question of the early decade 2000, I would say 11, 12 into 2014, 15. That was our question about 5 to 10 years ago. On that level, the way we looked into it is now on the level of the single 2009 algal bloom, becoming more evident if we combine taxonomy with metagenomics. Metagenomics meaning I take the DNA, sequence it completely, and look for abundant groups. And again, like in the taxonomy, we find in the metagenomes also Bacteroidetes and Formosa going from almost nothing up to a lot. So there's this change, this turnover, the substrate-based succession. If we now go into metaproteomics, the very same thing. You have the time points over here, and you have a massive change in the expression of particular proteins. So we now take the full proteome from the 0.2 to 3 micrometer fraction during the bacterioplankton bloom. These are the time points. We had 6 at that time. 1 in winter, an early bloom one, and late bloom ones. And you see how particular leukocyte hydrolases, plotted over here, take over. And this can now be linked. We can link this particular protein, this particular transporter or enzyme to a particular taxonomic group. And the result, once again, Bacteroidetes with particular transporters and particular K-zymes take over, followed by gamma proteobacteria, and within the phylum, a lot of changes among the different genera. Now, we go into 9, 10, 11, 12. And the way we look at that is due to Helgoland being a station, due to other people helping us. We have lots of data. So these are abiotic measurements typically done at each observatory for salinity, silicate, nitrate, nitrate, for each of the years. We can then look, and that's also data we don't do. We rely on our colleagues of the observatories. This is how a particular year, the year 2010, looks with respect to main diatoms, for example, and Phaeocystis, and Chatonella, and others. So we know exactly which type of phytoplankton is blooming. And that is then correlated with other things, like the bacterioplankton biodiversity, the cell abundances we count in microscopic ways with a technique called codfish. And it is combined then with metagenomics and metaproteomics. And what you constantly see is the major reaction is among glucoside hydrolases and other carbohydrate active enzymes, and among a transporter type to which I will come back in very much detail for the rest of the talk, the so-called starch utilization system, SAS-CD type transporters. I need to do a little bit of biochemistry so that some of you can catch up, because otherwise I would lose you here and you would not be able to follow. So please stay with me with a bit of basic biochemistry. So what you need to understand is that bacteria, of course, don't do endocytosis. They usually need to transport whatever they take up as food through membranes with transporters. Gram-negative bacteria, in particular the Bacteroidetes, have a type of transporter which is referred to as starch utilization system, SAS. C and D is a big pore, and the lid that is on top, a binding of the substrate, occurs to other outer membrane proteins, SAS-EF sometimes called, but these are polysaccharide binding proteins. The polysaccharide gets attached to the surface and more or less is then partially hydrolyzed on the surface and immediately transported through a big pore into the periplasm. So we talk about the outer membrane. Only in the periplasm, there's a full degradation of the oligosaccharide to mono D and trisaccharides, which are then frequently transported through ABC transporters into the cytoplasm, where further degradation energy production is happening. On the genomes, those things are recognized as co-localizations of transporters SAS-CD, indicated here as open reading frames, together with K-zymes and sometimes accessory enzymes that, for example, remove a sulfate group from a polysaccharide. So this is a polysaccharide utilization locus, a simple one. Now, let's go into one of the projects that happened in the second half of the last decade. It was published in 2019. Lennart Koppelmann and his PhD. He now used what was available to us based on several years of cultivation by Jens Harder and colleagues from North Sea surface water. We had some 53 bacterioplankton. I hope that the connection stays safe. We had 53 bacterioplankton isolates from the group of Flavobacteria. This is an order within the Bacteroidetes. So we stay within those gram-negative bacteria with that setup, the cell biological setup that I just explained. We did full genome sequencing of the isolates of high quality with the Joint Genome Institute in the US. We added the Gramella Forseti that was done with EU funding and some others, and then compared the number of polysaccharide utilization loci. These are indicated as green rectangles over here, and they could be annotated as the likely substrate I use. But this is a hypothetical nomenclature, likely utilizing laminarin or alginate starch or other alpha-mannans or what's that? A fructose containing sulfated polysaccharide, etc. But overall, we found some 400 pools, and we could say that isolates obtained from particles on average have more pools per genome than those obtained from surface water. That was one thing. And the second thing was then you now have not single genes that you look at, but you have loci. And those loci, once again, they are built with the transporter SAS-CD, maybe some regulation of the transporters, and then a particular combination of GH92. For those, you can predict it's likely alpha-mannosidases. There are more GH92, but there's GH3, predicting beta-glucosidases, etc., and many sulfatases. Based on that, you can say this particular single locus is potentially targeting a complex sulfated mannose-rich polysaccharide. But this needs to be verified. And a lot of the work done by our colleagues in current research is about validation of these predictions. The second thing that Lennart found out was if you were looking at the protein alignments of the transporters and looked at the predicted target molecules for those transporters, predicted based on the pull, on the co-localization of other genes, then there was a clustering of the transporters according to the substrate. So there's a substrate-specific clustering, both of the SAS-C and the SAS-D, which indicated that a proteomic assessment of transporters might be a good proxy for glycan utilization. Now, I told you in the very beginning, we know that bacteria remineralize biomass, algal biomass. But we don't know, even in the year 2021, what are the dominant polysaccharides through which this process is happening. We know that they are easily utilized polysaccharides like laminin, that's the starch of the diatoms. It's their storage molecule. But we also know that they are secreting particular molecules of almost, I would say, half-known structure, Fukuidan type things. And we know that they have cell walls that are very complex. And a part of the energy flow that goes from CO2 into biomass and back into CO2 is not understood on the glycan basis. And here, in the genomes, we have indications for the structure of those glycans, but those need to be verified. They need to be tested by glycobiology and by proteomics. So let's go to the next one. That's just the second topic right now. And that is, I now talked about that world that was cultivated. I talked about isolates. And the isolates, they are that part of the diversity that can be cultured in the laboratory in isolation. These are pure cultures. And in the environment, bacteria never live in a pure culture. They always live together with other bacteria. And if you just want to recall one important bit of information from the last talk, the average genome size of an isolate is 3 to 4 million base pairs. We now look into metagenomes. The years 2010, 11, 12 were different with respect to the type of algal bloom. You see the types of, let's say, phytoplanktons that we detected. You see that we then tried to do big metagenome sequencing. In 2010, 11, 12, overall 38 samples, some of them pre-bloom, most of them in the bloom, some post-bloom. In some years, we don't have a single bloom, but let's say a first and a second bloom. So we tried to be in the blooms and past the blooms. The way in which we worked was very simple. From a metagenome, you first do an assembly, and then the moment you have pieces that are long enough, so assembled pieces of some 5 kb are, for example, long enough, or 3 kb or 10 kb. You can look into Paymer frequency, so the genome language that is used, and the read frequency. And based on that, you can assemble or bin, let's say you have assembled sequences, you bin them into pieces that likely are coming from closely related strains of one species. So what we do is meta-genome assembled genomes. MEG is the word over here. So individual sequences assembled, binned, and then we look into the MEGs, and the MEGs, the metagenome assembled genomes of 38 time points, are now coded over here, once again as rectangles that are close or distant from others. and a mesh cluster distance of 95% average nucleotide identity indicates that these are likely strains of one species. Now, if we go into that, this is the image that we got from those 38 samples in 2010, 11, 12, color-coded. And these are the distances. So what it tells you is there's a limited number of recurrent genera of Bacteroidetes, some of them with cultured representatives. So whenever there's a name that is a full name like Aurantivirga or Formosa or Polaribacter, we have isolates. In this case, for the North Sea group 5, we don't have an isolate, or also for the UA16 family. And also, we don't have an isolate yet for Candidatus Procelliococcus, but that's a taxonomic trick where we try to describe valid taxa without having an isolate. But that's a separate talk and a separate discussion. But for you, the important thing is, whenever we look at North Sea off Helgoland in a spring situation, the likeliness to bump into one of 13, 14, 15 dominant Bacteroidetes groups is very high. Some of them recur every year. So that color coding, green, red, blue, is for the Cyclobacteria, but also for the Cryomorphaceae, also for the Aurantivirga, indicating that most often they occur in every year. Other groups like this particular Polaribacter was only found in 2010 and not detected as a dominant one. It will certainly be present, but it was not a dominant one in 2011 and 12. So, in terms of taxonomy, genome-based taxonomy, the diversity is restricted. So this is now a genome tree, and you see certain of the groups. Maybe you focus on Polaribacter, Aurantivirga, and Formosa as dominant groups for which we also have isolates. And maybe you look at others like Procelliococcus and some other groups, NS5, for example. Just look, these are recurrent mesh clusters, so metagenome assembled genome. What was the big surprise? Initially, the very big one for me, and the puzzling one, is the metagenome assembled genomes, which should, according to our statistics, be fairly complete. So those things are not closed genomes, but our statistics indicate they should be fairly complete. The average size of those is rather in the range of 2 to 2.5, and only in rare occasions up to 3.54, which the isolates have. So it seems to us as if there's a type of bacterium out there that can be cultivated with a rather big genome, whereas maybe as still a speculation, most of the cells that win in an environment have streamlined genomes might be auxotrophic for certain cofactors, vitamins, or other things that are provided by other bacteria, and therefore they cannot grow in pure culture. That's the first one. The second one was for us, I would say, even more surprising, and that is we looked for polysaccharide utilization loci in the metagenome assembled genomes. With a simple algorithm, we looked for a window of 10 genes, and in those 10 genes, there should be 3 that are either transporters of the Sas-CD type or sulfatases or casezymes. So would fit within the canonical definition of a pool. And then we looked into what can be found in there. And we found some 1,200 Sas-Cs and about the same number, 1,300 Sas-Ds in our metagenomes. Then we looked into a reduction based on identity, and we found only 130 different type of transporters to be abundant in our metagenome assembled genomes in the 0.2 to 3 micrometer fraction of spring phytoplankton blooms of Helgoland. So just that you understand the window. We look into one ecosystem which we recurrently sample at an observatory. So there the diversity is limited. And how did the diversity look like? And this is now looking at the SASD. And what we do is we just look at SASD and predict the substrate as we did before based on colocalization of K-Zymes. And we find exactly the same substrates as indicated in the pure cultures, as indicated in the genomes of the pure cultures. So what you recall over here is the laminarines, the alpha-glucans, the alginate, some mannans, some sulfated xylans, etc. So that's what we found out there. And we found certain families, which you by now recall. So Candidatus Procyliococcus, Aurantivirga, Formosa, Polaribacter. If we look into the more simple substrates, you see that all of the abundant groups can use the simple abundant substrates like the beta-glucan laminarin or the alpha-glucans. So most of the cells go for the abundant substrates. In contrast, if we look for the more complex, like let's say mannans, sulfide xylans, or alginate, more complex ones, only few ones. In this particular case, the Formosas, Aurantivirgas, and Polaribacters in particular can use those substrates as well. And as a summary, you could more or less say the SAS-C confirms the SAS-D finding. And we have groups like the Cryomorpha-C or the Candidatus Procyliococcus, which can only use one, maybe two substrates, the laminarin and maybe an alpha-glucan. Whereas others, and I indicated Formosa, Aurantivirga, Polaribacter, most likely have a more diverse full spectrum, indicating a more diverse glycan utilization pattern. So those things have overall 6, 7, sometimes 8 pools. But be careful, within the genus, there's also specialists. So there are other strains or species within the genus that have a much more restricted spectrum of polysaccharides that they can utilize. So that's the current situation. Overall, I would say the diversity on the dominant groups is, interestingly enough, not tremendously complex. So what is currently going on? What are we doing in 2021 and 2022? The most important one is, of course, we have now, I would say, a collection of polysaccharide utilization loci. We have a collection of isolates. We know what the abundant metagenome assembled genomes are, but how gene expression is happening over the course of a spring bloom is has been, let's say, the target of frequent sampling in 2020 with more metagenomes and more metaproteomes than ever. This is a project funded by the German Research Foundation right now for another 2 years. And we also will start to look into those bacteria that are in the 0.2 to 3 micrometer fraction and those that stick on particles. Just assuming that the leftovers of the phytoplankton are most likely more complex in composition with respect to polysaccharides, but also with respect maybe to glycoproteins or other combinations. And in this particular case, we then need to look in the larger than 3 micrometer. Plus, we do a lot, as I indicated, in verifying the substrate specificity of a locus. So really confirming for an organism that we don't know that the pull is most likely indicated a class of substrate, but now we need to nail down the substrate. So the question really becomes the predicted substrate. Can we show that an organism, a bacterium that is potentially still not cultured, can that particular utilization of a new substrate, can that be shown? And to that, we go into the third part of the talk, I would say, and that is now a microscopic one. Let's just shortly recapitulate the type of transporter that I was describing, the SAS-CD type of atom B-dependent outer membrane transporter. So I talked about that particular situation. And why do we focus so much on this particular transporter type? Now, the most important sentence right now, and I should have done that maybe already 15 minutes earlier to keep your attention, is up to 20% of all protein that is present in the 0.2 to 3 micrometer fraction. So in overall bacterioplankton, in the free-living bacteria, up to 20% of the protein is of the SAS-C and D type. So the bacteria, a heterotrophic bacteria, first of all needs energy. And it can only get polysaccharides for their own utilization if they invest heavily into the transporters. So almost more abundant or as abundant as ribosomal proteins, this particular transporter type is the dominant one in the metaproteome of a spring phytoplankton. And the trick is the following. If you have invested into that uptake system, you monopolize a particular polysaccharide by binding it, grading it into an oligosaccharide that cannot be used by everybody because it is not released from the cell surface. Then you put it into the periplasm, and only there you do the degradation to monosaccharides, which could be used. Glucose can be used by many, many bacteria. But those oligosaccharides can only go through that channel. So you monopolize a main energy resource in the surface wall. And if that is true, it should be possible that you use a fluorescein labeled polysaccharide. And we tested that in 2012 for the first time with a microscope in a transatlantic cruise, where we utilized fluorescently labeled polysaccharides. And we found those types of pictures. You find a staining that is a circular one surrounding the cell. And that is reminiscent of a staining of the periplasm. So the fluorescent stained polysaccharide, oligosaccharide, goes into the periplasm. It is bound to the surface as well. And that's why you get that type of staining. Now, if you go into the whole setup, you will understand that this is now amazingly simple. The only thing that you need is, for example, a laminarin, which you can order from Sigma as a purified polysaccharide. You label it covalently with fluorescein. You put that into surface water. I said we were with the with the Ross, with the English ship in the transatlantic situation. We took water freshly from the CTD, added the substrate, waited for an hour, a day, and then had controls, and then the staining looked like that. And the staining is a staining of the periplasm that is already evident sometimes after an hour, sometimes you need a day. You can look for the number of cells that are present on that microscopic field. These are 1, 2, 3, 4, 5, 6, maybe 7. 3 of them are Bacteroidetes. And those 3, this, and this, have taken up laminary. There's one more cell over here, there, and there that is not a Bacteroidetes, but takes up laminary. And then you can have a combination of a stain for taxonomy with a combination of a stain for laminary. Based on that, we can now predict that, unlike written in the textbook, where the assumption was that bacteria need to excrete exoenzymes in order to degrade polysaccharides into monosaccharides, which would be public goods also utilized by bacteria that don't have glycoside hydrolases, that don't degrade polysaccharides. So a system that is leaky and a system that is definitely also out there. So there's no doubt that this, let's say, mechanism exists. But there's a second mechanism where bacteria invest a lot of the genome into binding to the outer membrane of a large polysaccharide and uptake into the periplasm. This way, behaving selfish, so monopolizing an energy resource for themselves. So that's a new mechanism. That this is really a periplasmatic uptake. You can visualize on planktomycetes. Planktomycetes are bacteria that are also in a setup gram-negative, and they have a huge periplasm, referred to as paruphoplasm. So in this particular case, you see the DNA of the cells, the ribosomes of the cells, and in a huge periplasmic space. And this is also stained, in this particular case, with the chondroitin sulfate. So that's a more complex sulfated sugar, also labeled with fluorescent. So what I showed you is this is really a periplasmic uptake. Now I come to the end of my talk and would like to do a couple of conclusions. So the first is it's a golden time for all of us who would like to find out more about complex marine systems. The metagenomics in particular is amazingly powerful because sequencing has become so cheap, and we can learn a lot, in particular on the important things of biogeochemistry, the marine carbon cycle, for example, and how bacteria catalyze them. We can learn a lot on the diversity, which is interestingly in the coastal seas, in certain situations limited. There's a long discussion right now whether there are millions or billions of species out there, but in a spring situation of Helgoland, there are certainly only a couple of, maybe dozens or 100, which is really important. We can predict, and I told you, the sugars by now. These are not validated. We might at this time see more than a glycobiologist or sugar chemist can see in the genomes, but we need to collaborate transdisciplinarily with those specialists to validate our hypothesis. Third one, we need to consider new uptake mechanisms. So even the most basic understanding on how bacteria interact with polysaccharides in the ocean has changed, I would say, and we need to find out how much of the new selfish uptake mechanism is contributing to element cycling and how much the more traditional exoenzyme-based one is out there. So we need to be quantitative in those microbiome studies in order to advance our understanding of diversity and function of marine microbes. So what are we currently doing as an outlook? First of all, we need to find out whether the Helgoland microbiome is a good proxy for coastal seas of higher latitudes, so of the temperate and polar situation, because the blue oceans are certainly quite different. Blue oceans meaning tropical, subtropical ones. We also need to integrate that type of research. So getting fractionated biomass, isolating DNA, doing a lot of, I would say, comparative 16S sequencing and metagenomic sequencing into the observatories, because global change will impact the way bacterioplankton and phytoplankton is interacting. Just as a last remark, you always have to recall that about every second breath that you take is coming from oxygen generated in the oceans. And about 1 out of 5 of these breaths that you take in a regular day comes from a diatom photosynthesis, which is creating a biomass, which is subsequently mineralized by bacteria. So that just gives you a reflection on how big the element cycles are that we are currently addressing. And Helgoland might be indeed, I think we have indications for that, a good proxy for temperate and polar seas. And looking at those things on a decadal scale by, I would say, enriching the observatories with respect to size fractionated sampling of bacterioplankton. Some observatories do phytoplankton, many do zooplankton and fish, etc. But we need to get also into the microbiomes, into the bacterioplankton. And on the long run, I did not mention it at all today, but on the long run, we would also need to sample the varroa, so the sum of the viruses that are in a particular water sample. Okay, that's enough for today. I thank those of you who stayed with me. I would like to thank a couple of people who worked for many years with me, Bernhard Fuchs, an ecologist, Jens Hauder, who isolates Bacteroidetes and does other things. Three very, very good PhD students who now either are postdocs or went out of science. So Lennart, Karen, Greta. Arno Tilling was our pioneer in bioinformatics. And all of what I told you would not have been possible without our colleagues in Helgoland and Greifswald, too many to really name them over here, but they are on the co-authors on the publications. There was a lot of funding over 20 years. The current one is the most important one for now is the German Science Foundation, EFG, and the project POMPU, a Forschergruppe 2406. The backbone is always Max Planck Society. I had a lot of funding from the Joint Genome Institute and a lot of funding by EU and BMBF in the not the last couple of years, but before. And right now, I think that's all. I thank you, and I'm ready for taking some questions.
Thank you, Rudy, for this excellent presentation. If there are any questions, please indicate in the chat so you can see who it is, and then you can ask the question. No? Seems as if there are no questions. Well, if not, then I would like to thank again Rudy for this talk and for collaborating with us. It is great to see you again, and we hope that in better circumstances we can meet again. Oh, there's Nick. Nick has got a question.
Okay.
Okay, Nick, you can talk. You can talk.
Thank you very much for a very interesting discussion, Rudy. I was very interested in observatories at the moment. We're involved in that as well. And particularly, I thought your final comment was interesting. And I was wondering if you'd elaborate a bit more about the collaborations that you have ongoing around microbiomes as a whole, or whether the other observatories than the Helgoland one.
Yeah. So first of all, in Germany, we have this DRM, the Deutsche Allianz für Meeresforschung, which is just forming. So it's the German alliance of the different research institutions doing marine biology, also the universities. And over there, we will have a mission, which is a mission for the protection and sustainable utilization of the German seas, what is the North Sea and the Baltic Sea. On that level, we are currently pushing for integrating regular sampling of size fractionated bacterioplankton. Initially, not yet the varroa, because I think that's something that is further along the line, because the sampling is not so easy. But that will be, let's say, I'm optimistic as always, that will probably be integrated into the observatories in the Baltic Sea and the North Sea, and also some of the regular transect journeys. I know that, let's say, the English, the French, and some others have started those observatories to be complemented or let's say, made ready for the future by also sampling size fractions that are bacterioplankton size fractions. I'm personally not so much involved in the collaborations, but maybe Nicholas, you could give me some update. I know that in Roscoff there's something going on. There's the Plymouth time series, etc. Are you aware of other stations that I'm not aware at this time where they already do that on a more regular basis? Are they in assemble course?
So there's a big observatory also in the Bay of Naples, which is one of these augmented observatories. So they're starting to integrate imaging as well. The same thing is happening in the south of France. So those are the ones that I'm aware of. But off the back of Assemble Plus and the joint research activity we had on genomics observatories, we're looking to put one in place for EMBRC as well. So we're trying to do that this year. Hopefully we'll go online in the first half of the year, where we'll start sampling. We're not doing viruses yet either, but I noted your comment on the importance of this, and we've noted this as well. But it's always the issue of getting things started and deciding which fractions are you going to do now and what you're going to do later.
Let's say we should not underestimate the importance of that. Let's say that we can do it right now, but it will be complicated. So a sampling for a virome these days is not standard. You need still quite a lot of water. So an automatic sampling where you have a fixed sampling would most likely not give you the biomass that you need. And size fractionation is also not so straightforward. So what I anticipate to get the funding is really that we try to go into at least a weekly sampling at selected observatory that go on annually for a decadal, let's say, perspective. That means it would be a really, really big exercise. In particular, let's say, if you do that in a network of things, you would potentially have hundreds to thousands of samples per month. Then you just upscale it. And the bioarchive would be the most important thing. You put things at -80 and you put them away for later examination, because I'm very optimistic that sequencing will continue to become cheaper and cheaper and more straightforward. But even today, it is amazing what you can do with Illumina, which is extremely high throughput, and in terms of length with PacBio. So we are currently doing a lot this year and last year with PacBio as well, because you get long reads. So the potential is out there, but it needs long-term funding, at least for the bioarchive, and then a systematic approach in the EU to get towards the microbiome and the virome. Just considering COVID-19, I'm not claiming that we would have detected COVID-19 in the surface oceans. That's not what I claim. But it shows you how important the invisible things are for our sustainable life on the planet. So I'm not claiming that we will prevent future pandemics, but the impact that the pandemic has is certainly that people realize we need to have an eye on our surrounding in the environment, but also in, let's say, in the apartment and in the institute with respect to microbiome and virome.
Thank you. If I may just respond to that, Lino, before you let Conan ask his question, I'd just say that we would be very happy to speak to you about the methodologies on the virome, because I think that's going to be interesting. And so I'll be in touch with you. But I also think that you touch on something very important, which is the long-term sustainability, which is where I think we're all struggling with the biological observation. This is very difficult to obtain, and it would be good if we could all sort of come to a joint strategy on this to try and get back to having long-term observation and it being, leading good science, which it's had in the past. Thank you very much for your time in your answers.
Thank you.
Thank you. So Colin Brownlee, can you ask your question?
Yeah, it's just a quick question. Rudy, very nice talk. You mentioned that many of these species are unculturable. So I just wanted to ask you a general question. What do you think the prospects are for culturing in the future?
So that's a very interesting question. So we are just through massive attempts of cultivation. So we finished two, I would say, multi-year cultivation PhD studies. One was published in 2020 by Carlota Alejandre, 5,000 strains on plates. And on the plates, we always find the same things. So it's little progress that we make in terms of getting some of those NS3, NS5 groups that are typical of the North Sea and which turn out to be not culturable at this time. Now, one problem is that with a dilution culture, so that you don't go on an agar plate, you get other bacteria to grow. Also some of the new ones, because dilution cultivation has been more limited in the past. So you get some more, but you're still left with lacking some groups, in particular those with the smaller genomes. And we expect, and this is an unproven hypothesis, but let's say we see that in the metagenome assembled genomes, which are inherently difficult because they are incomplete. But the prediction on completeness is 99%. With the PAC biosequencing, we will be able to get fully closed genomes from the environment. And whatever we see over there, the genomes are small and the organisms are multi-auxotrophic. So that means they lack the ability to do important things and they cannot live in pure culture. So my take on that is the marine microbiome is so highly evolved that dominant organisms, those that can rapidly grow on a changed environment based on a algae bloom, have small genomes, because then you are not so heavy. You are small, you can quickly grow, and based on that, you will win. And you do that just assuming that a particular cofactor, vitamin, or something else, an amino acid, that you cannot do yourself, that you can, let's say, not synthesize, that you have it in the surrounding water and that you just need a transporter for it. And that type of auxotrophy will, of course, prevent any attempt of pure culture isolation. It will never work unless you can formulate a growth medium which has all of those more complex components present in the right concentration, and it's a mix. So this will be difficult. But on a long run, I dislike the term non-culturable. It's yet uncultured. That's an important difference, because in nature, those organisms have no problem whatsoever to grow. They just hesitate to come to the lab and to go into isolation.
Great, thank you.
Thank you. It seems that there are no more questions. So again, Rudy, thank you very much. And thank you everyone for listening. And we'll come back at 11:30 Central European time for the next session. Thank you very much.
Bye-bye. Thank you.
Thank you.